Hi, everyone. Good evening. Thank you for joining the NATF online support group. We are here this evening with Dr. Christian Ruff, who is a cardiologist at Brigham and Women's Hospital, as well as the director of general cardiology. And he is an assistant professor of medicine at Harvard Medical School, and he is also one of the NATF board members. And we're really fortunate to have him here with us tonight to talk about a topic we get a lot of questions about. Genetics with blood clots, and it's in particularly very important for those of you I know who have children. You have a lot of questions about this. So all the lines are muted. If you have a question, the best way to get it in is to enter it in the chat box, which is on the toolbar, the GoToWebinar control panel. If for any reason that's not working for you, you can email it to info@natfonline.org. And with that, I will turn it over to Dr. Ruff.
Great. Thank you very much, Catherine. It's a pleasure to speak with you guys tonight on a topic that I think is very exciting, particularly in this day and age. We're going to talk about genetics for sure, but more broadly about a question that I'm frequently asked, and I actually do a lot of genetics research with respect to cardiovascular disease, and that's obviously when you either have an event, a clot, either a heart attack, a stroke, or a DVT, or you have a family member that has a clot or a thrombus, is both the patients want to know, can you get a genetic test or another test, a blood test that will tell you what your risk of having another clot is? And also, is this something that's inherited that people in your family, children, parents, sisters, brothers should be tested for? And so I'm going to talk about this really quest for the Holy Grail, and that can a single blood test determine your risk of cardiovascular disease and having a blood clot? And we'll focus on the genetics as one of those single blood tests. But basically, any blood test that you order, whether it's measuring genetics, which we traditionally think of as DNA, or whether we're measuring proteins on the blood, what we're really trying to figure out is, can we find the perfect biological marker for disease? And when you're looking for a biological marker, there are definitions for that. This is one from the World Health Organization. It's really any measurement that reflects an interaction between a biologic system and a hazard, be it chemical, physical, or biological. In this case, say we're talking about venous thromboembolism, is can we measure something that's going to reflect the risk of forming a clot, either in the deep venous veins or the pulmonary arteries. And you can measure it in a variety of ways. It could be a genetic test, it could be a functional test, a physiologic test, biochemical. It's really any measurement that gives you insight into the biology of forming a clot. Now, we know if we take cardiovascular disease in general, cardiovascular disease, one of the major drivers of the morbidity and mortality of cardiovascular disease is really blood clots. And you could form a blood clot in the heart, and we call those heart attacks. You can have a blood clot that forms in the heart and goes to the brain, and that's a stroke. And we know patients with atrial fibrillation are at risk of having a stroke. And of course, you can have a blood clot in the veins, which could form a deep venous thrombosis or pulmonary embolism. And it turns out that the biology of forming clots, whether it's in the arteries or in veins, tend to be very similar. And many of the same patients could be at risk for having multiple of these types of clots, a heart attack, DVT or PE, and a stroke from atrial fibrillation. That's why the North American Thrombosis Forum really deals with all three of these diseases, because forming a clot, the treatments are often very similar, but also the underlying biology that drives people to form clots also has a lot of shared characteristics. So if we want to know our risk of having a clot, there are a variety of different things that predispose a patient to having a clot. Obviously, there's evolutionary drivers for clots. Obviously we clot because our ancestors, if they were bitten by a tiger or some other predator, would need to stop the bleeding. And so we're physiologically adapted to form clots. Now, obviously, that can become pathologic, and some people are too predisposed to having a clot, but forming a clot's important. And just like every other factor, there's variation in what people inherit. We know that gender plays a role. Men are more women that have blood clots than women. We know that there are things that are inherited in our DNA, which I'm going to talk about. And we know that certain families may be at risk for having a clot more than other families. But we know environmental exposures are often also very critical. That could be smoking, which we know is a big driver of forming a heart clot or a leg clot or a lung clot. Oral contraceptives, also very important environmental trigger for DVT. We know cholesterol, particularly important in heart and brain arteries. Blood pressure, which is actually important for both arteries and veins. And we know activity. Obesity is a big driver of forming clots. It's thought that obesity actually causes a low level inflammation throughout our body, and that low level of inflammation predisposes us to forming clots in arteries and veins. And we know other diseases that are commonly associated as we age, such as diabetes, also may increase our risk of having either a DVT or a heart attack. We do know that, let's not talk about even genetic tests, but we have pretty good ways of estimating people's risk of having clots. This is an example for a heart risk calculator for heart attacks. This is by the American College of Cardiology and the American Heart Association. You could find this on any website, where you could simply enter your age, your gender, your race, blood tests, total cholesterol, good cholesterol, your blood pressure, whether you're diabetic, whether you smoke. And if you just enter that in for each patient, we do this in clinic every week, you can estimate what someone's 10-year risk of having a heart attack. And so that patient that I entered for here, their 10-year risk was 44.6% of having a heart disease and stroke over 10 years. And that risk is fairly elevated. So the guidelines would recommend that this patient would be on a high intensity statin and would have more aggressive control of their blood pressure. The same thing is true for DVT-PE. When a patient has a venous thromboembolic event and they're obviously concerned about having another clot and whether they need to be on lifelong anticoagulation, there are risk scores where you can go to online, whether you're a patient or physician. One of the common ones is the DASH prediction risk score, and you could put in, some lab tests. What's your D-dimer, which is a protein in the coagulation cascade? Is that elevated? If it is elevated, you're at higher risk of having recurrent clot. What your age is. Again, if you're a male or female, and we know men and older people are at higher risk of having a clot. And as I mentioned, an environmental trigger here, hormone use for females, particularly oral contraceptives or estrogen replacement. And you could get what someone's annual risk of having a clot. In this case, the patient's annual risk was almost 11%, and that's per year, so that's a pretty elevated risk as well. And for this patient, we'd probably recommend that they continue on lifelong anticoagulation. So we're not talking about genetics here, but we already have clinical risk scores that incorporate clinical variables such as age, gender, et cetera, as well as specific blood tests, in this case proteins, that we know to be involved in the disease process. So for example, we know cholesterol, very important in the risk of heart attack. So we generally measure cholesterol for heart disease. And we know that coagulation cascade, or the ability to form clots, is very important in venous thromboembolism. And as I mentioned, D-dimer is a common protein that we can assess. But of course, these clinical risk scores are great and we use them routinely, but we still, both physicians and patients, are really on the quest for the Holy Grail. And the Holy Grail is, wouldn't it be wonderful if just a single blood test could gauge cardiovascular health? So that we didn't have to do all of these risk scores, that we could simply measure one thing, and based on that one test, we would know how to treat a patient. And so now, what would that one test be? Well, we know that proteins in the blood are the sort of machinery of how all of the cells act. And we know that there are many different proteins in the blood that are involved in forming a thrombus or a clot in an artery. And you don't have to pay attention to the particular names of these. They're all quite funny. But there are various disease pathways, both in inflammation, in heart stress, in remodeling of vessel walls, metabolic proteins, and there are dozens, if not hundreds, of proteins that all interact with each other, both to increase or decrease someone's risk of having a heart attack. So we can measure a bunch of these different proteins. And as I mentioned, we're already measuring some proteins. We're measuring cholesterol, we're measuring D-dimer. So we're already measuring some of these in patients, but obviously there are hundreds of these proteins we could potentially measure. And we're always thinking about, is there some new, better protein that we could measure that would give a better gauge of someone's risk of having a recurrent clot. But I wanted to talk to you specifically today, because there's been a lot of enthusiasm in the Human Genome Project, is could we measure or study someone's DNA to tell us if someone, maybe even as early as a child, are they at risk for having a heart attack, a stroke, in this case a DVT, or in someone who's had a clot, what is their risk of having another clot? Could we safely discontinue a blood thinner and know that their risk of having another clot is lower, say, than someone who inherits a DNA variation that predisposes them? And first, just to give you a little bit of background, because you'll hear a lot of terms when you follow this literature, is that you'll often hear of the genome. And the genome is basically the entire set of genetic instructions encoded in DNA found in a cell. So that's everybody inherits a genome, and the genome is stable. You're born with it. Once your parents mate, you have a fixed set of DNA, and everybody has their own individual genome. And then genetics, which we're talking about today, is the study and function of single genes and how it interacts with other genes in the environment, because the genes don't necessarily dictate whether you have a disease or not. Often they may predispose you to a disease or they may protect you from a disease, but there are a lot of other important environmental factors that may determine whether that given individual actually develops this disease. And then, of course, there's genomics, which is not really just studying single genes, but studying various genes that may interact in a given pathway. And this will become very important later in the talk. So we're all born, if we're born with sort of normal chromosomes, with 46 chromosomes. They come in 23 pairs. One is the sex chromosome, so you're either XX for female or XY for male. In your whole genome, so every person is born with about 3 billion base pairs, so that's a lot of base pairs. There are 4 base pairs, A, T, C, and G, and different combinations of them make up your 3 billion base pairs. It's interesting, though, when we talk about genes, so genes are the regions of the DNA that encode for a protein. And that's what a gene means. Gene means it's an area of DNA that gets basically transcribed and then translated to a protein. And as I mentioned, the DNA itself doesn't really do anything. The DNA just provides the blueprint for something to happen. An example might be, say you're building a house. The DNA would really be the architect's plan, the blueprint. And then the blueprint, though, has to be made into a house somewhere. And the blueprint, basically, the people who carry out the information in the blueprint, say, are the contractors. And the proteins in the body are really the contractors. So the DNA gives the information to say what proteins need to be made, and those proteins are actually the workhorse of the body. And so interestingly, although you have 3 billion base pairs, only 1% of your entire DNA actually encodes for genes. There are only 20,000 genes in the entire human genome. What about the other 99% of your DNA? Well, it turns out that a lot of that DNA actually helps regulate whether genes are turned on or off. And to be honest, a lot of the other DNA, we have no idea what the role is, if any. And so we're still learning very much about what all the rest of this DNA is. But I think people always find it interesting that when we talk about genes and genes causing disease, we're really talking about 1% of the genome. Now, what I think is also very interesting is that out of those 3 billion base pairs, so 3 billion sequences of DNA. 99.5% of all the DNA you have in your body is identical to every other human on the planet. So what makes each of us unique as a human individual is really restricted to changes in DNA that is in less than 0.5% of your entire DNA. So almost all of our DNA is completely identical. And the variation that means that you're a little different from your mother, father, neighbor, people who live in another part of the world, is all changes in a relatively small amount of your DNA, 0.5%. And most of those differences in the DNA are single base pair differences between people. And again, we don't have to go through the details. It's really for geneticists. But the entire revolution in genetics has basically been predicated on these things called SNPs, which are single nucleotide polymorphisms. And so every 300 base pairs, there is a change in someone's DNA from person to person. Either, say in this example, person 1 has an A at that position, person 2 has a G, person 3 has a T. So the single base pair change is what underlies human variation. Now, these changes could be entirely meaningless. So say one of these changes occurs in an area of the DNA that doesn't do very much. Then it really doesn't matter what you change this base pair to. It doesn't really affect that person's risk of having a disease. But say this change occurred in a gene. Remember, those genes are only 1% of the genome, but they're very important. Well, this change may make the gene more active, or it may make it less active. And that change in the gene may affect someone's risk of having a disease. Say this is an important gene, and this change, say from going an A base pair to a G pair, makes the protein ineffective. Well, then that person will no longer have this critical protein, and they'd be more likely to have a disease. And so that's the whole basis of human genetic variation. And our ability to categorize these changes throughout someone's entire DNA has led to the ability to detect genetic diseases. And so we're going to talk specifically about venous thromboembolism. And we've known for a long time that there certainly is an inherited component for VTE, that there are certain families that seem to be at higher risk of having a VTE than others. And what are some of the clinical features that make us suspicious that there may be a genetic component to the venous thromboembolic event? Certainly having a clot at a young age, less than 50, we like to say, especially if it's not in the setting of some major catastrophic event. Like obviously someone had a horrific motor vehicle accident, was laid up in traction for 3 months. That's a pretty big stimulus for having a VTE. But say someone, were on oral contraceptives or at a minor surgical event and had a clot in their 40s, that would be a little bit unusual for a minor provoking factor. Certainly patients that when we take a family history, they have a strong family history. That's usually multiple first-degree family members that are infected at a young age. So it's not, I had a grandmother who had a clot when she was 65. It's like, oh, actually, my paternal grandmother had a clot when she was 46, and then my mother had a clot when she was 42, and I had a sister who had a clot when she was 35. So that's multiple first-degree family members, and they all had clots at a young age. Certainly people who have recurrent clots, particularly when they're young. So if you have a person that's had multiple clots, especially if they've been on a blood thinner, that's usually some inherited component. And then there are particular VTEs that are in weird sites. Most VTEs are in the legs, sometimes the arms. Obviously, the clots in the legs can go to the lungs. But sometimes people have clots in weird veins, like in the gut or in the head. That's unusual. Patients with VT in those areas make us think that there may be an inherited component. And it turns out that over the years, we have detected different proteins and genes that we know predispose patients to having a clot. And these are the 5 most common ones. Antithrombin deficiency, or AT, protein C deficiency, protein S deficiency, factor V Leiden, which is the most common, and prothrombin gene mutation. And again, the names aren't important, but just to let everyone know that there are 5 well-described genes that encode for proteins that we know that particularly deficient versions of them predispose patients to having a clot. And we know that if you have people who have one of these mutations, that it increases their risk of having a clot somewhere around, 3 to 7-fold, with one exception, antithrombin deficiency. So it might seem obvious, like, oh, these factors increase the risk of having a clot. Shouldn't we measure them in everybody who has a clot? And if they have one of these or multiple one of these, shouldn't we then say, okay, you're at really high risk. You need to be on a blood thinner forever, and maybe we should measure these same genes or proteins in other family members to say, if you have one of these, and you haven't had a clot, maybe you should be on a blood thinner prophylactically so you won't develop a clot. It turns out it's not that simple. Although these particular genes in isolation increase your relative risk of having a clot, if you look at people who have these mutations, their chances of having a clot per year are vanishingly low. So you see here the incidence per year are well less than 0.5% per year. In fact, usually 100 times less than that. So for example, I can go out and measure these things in all of the people in the United States, and I'm going to get hundreds of thousands, millions of people who have one of these mutations. But their risk, that individual patient's risk of having a clot, is vanishingly low. In fact, so low that if you do the statistics for this, that even if you found one of these in someone who didn't have a clot, that their risk wouldn't be high enough to warrant them being on a blood thinner. And what I think is even more surprising for patients is that even if you took a patient that had a clot and you said, okay, you've had a clot, and I measure these things, and let's just give factor V Leiden because it's the most common, and you say, I found a variant, that you inherited a variant for factor V Leiden. And even if you took those patients and you said patients who had factor V Leiden, everyone had a clot, and you said, I take half the patients who have factor V Leiden, the other half who didn't, and I follow them for 10 years. Their risk of having a second clot is actually not statistically different, such that I couldn't use it to say, actually, the patients without the factor V Leiden or normal factor V Leiden, I'm not going to give them a blood thinner. But only the patients with the factor V Leiden are they at high enough risk to give a blood thinner. So although these individual genes increase the risk slightly, they don't increase the risk enough that you can make any treatment decisions whether to discontinue an anticoagulant or not. And even if you screened a family member, so you had a patient who came in, they had a clot, you measure these and you say, you have factor V Leiden or prothrombin gene mutation. Even if you screen their children or their sisters and brothers, even if you found them to have one of these variants, it doesn't increase their risk enough that you would give that patient who never had a clot a blood thinner to prevent the clot. And so this has been one of the challenges. And so the recommendations regarding genetic testing has actually been pretty muted with respect to venous thromboembolism. And there are a couple of important caveats for people on the call to remind you when we do and when we don't measure these genetic tests for recurrent clots. First of all, you should never have any of these tests done at the time that you've had the clot because they're entirely unreliable. And in fact, you can't actually reliably measure these things when someone's taking a blood thinner. So really, if you're going to measure these tests, the only time you can measure it is once they've completed anticoagulation for whatever clot that you were treating them for. And we really generally only consider testing in very specific situations. I'm going to review them on this slide, because the bottom line with respect to genetic testing for venous thromboembolism is actually pretty simplistic at this stage. It's that most patients with venous thromboembolism do not require thrombophilia testing, which is basically the genetic testing for having a blood clot, since the results will not affect management. And that's really the key question. If you are going to have a test done, you should only have a test done if your physician gets back the test and it changes how they treat you. But if I'm not going to give you a blood thinner, or I've decided I'm going to give you a blood thinner, and the genetic test doesn't change your risk profile based on your clinical characteristics, how old you are, where your clot was it associated with a provoked factor or not, then why am I ordering the test? Because I'm not doing anything different with that information. And the only time testing, at least in the guidelines, is recommended is only in younger patients who have a clot, particularly less than 50, with weak provoking factors. So again, not some big thing that we know is at risk for having a blood clots. So young patients with a strong family history, or they've had 2 or more clots. So that's a pretty small list. And I got to tell you, I mean, even being in Boston at Brigham and Women's Hospital, Harvard Medical School, we see a lot of thrombophilia testing, both within our hospital, but in community hospitals and other hospitals throughout Boston, Massachusetts, and actually throughout the country. And it's not clear what people are supposed to do with that information. In fact, it causes a lot of confusion. Now, this seems like I'm being pretty down about genetic testing. And I don't mean to be. And actually, I spend a large chunk of my time doing genetics research, as I mentioned, at the Broad Institute of Harvard and MIT. So I love genetics, and I think it plays a very important role in our future in providing personalized or precision medicine. I just think the current testing for venous thromboembolism right now is incomplete and limited and doesn't have much clinical utility. But what does the future have in store for us? And first, I would like to say that one of the issues about genetic testing is people really don't understand what we're talking about when we talk about understanding how genetics predisposes to disease. When you hear genetic testing, you're generally thinking of single gene disorders that predispose particularly young people, even kids, to horrible genetic diseases. The example is rare diseases like cystic fibrosis. So cystic fibrosis is a rare disease. Cystic fibrosis is caused by a mutation in a single gene, just that one gene. So if you have a normal, and the gene is called the CFTR gene, if you have the normal CFTR gene, you will not inherit cystic fibrosis. If you have a mutation, in this case 2 mutations in CFTR genes, you have an abnormal gene, you will develop cystic fibrosis, which is a devastating disease that causes children to have sticky and thick secretions, both in the lungs and the pancreas, et cetera, and often can be fatal unless someone has a lung transplant. So that's a single gene disorder where that gene, if it's mutated, basically causes disease. But those diseases are really rare. I'll give you some other examples like Huntington's disease, cystic fibrosis, et cetera. Most diseases that we take care of in medicine, and they include cardiovascular diseases such as heart attack, stroke, venous thromboembolism, are common diseases. And there isn't one single gene that causes the disease. In fact, there are multiple genes, and each gene doesn't have this huge effect like the cystic fibrosis gene, where basically, like, if you have a problem with the gene, you have a 100% chance of having the disease. Most genes, say, that are involved in venous thromboembolism, having a mutation in just one of the genes, that may only increase your risk by 1%, 2%. But you have to look at all of the different genes that are involved in that effect and sum them all up, because there may be dozens, if not hundreds, of genes involved in common diseases. And this is an example from coronary artery disease. And coronary artery disease was really the first common disease that we said, actually, there's not 1 or 2 genetic variants. There are, and this was from 2015, there were 60, now there's over 100. So there's over 100 genetic variants that we know can influence the risk of someone having a heart attack. And we're starting to do these studies now in venous thromboembolism. Now, each of these genes here showed on this wheel, they don't increase your risk by 80%, 90%. They increase it by 1%, 2%. And it's only by measuring all of these genes and adding all of their effects together would we understand if a patient's at higher or lower risk of having a disease. So we can't just measure those 5 genes that I told you about that we know are associated with venous thromboembolism. We need to measure dozens, if not hundreds of genes in order to develop a genetic risk score to predict someone's risk of having recurrent venous thromboembolism. So it's not that we can't use genetics to predict risk, it's that we haven't yet discovered all the genes we need to predict the risk of having a venous thromboembolic event. And the one thing I'd also caution you is that genetics doesn't operate in a vacuum. Then there's a seduction of reductionist thinking, where the sum of the parts equals the whole. So we could just look at like these individual genetic factors, and then we could figure out the bicycle. If we looked at the brakes, if we looked at the shocks, if we looked at the wheel, we'd understand the bicycle. But you can't reduce the human body to parts because basically you no longer understand the meaning of that individual's risk of disease. And it's important to remember that biological systems are very complex. Genes don't operate in a vacuum. Everything is connected to everything else. So in order to explain one factor, we need to understand all the others and the connection between them. So the answer is that genetics really has come a long way, and we're at a precipice where the computational power for genetics is just exploding. So our ability to measure every single base pair and every single patient we see is actually a reality now. But that's only part of the story, because we now are able to accumulate all sorts of data in every individual patient. So not just their DNA, but their blood tests for proteins, their imaging, their clinical characteristics, their family history, etc. And this is the era of big data. And this is actually a time not to be pessimistic, but actually quite optimistic. And for example, I like to tell people, let's go back to a space analogy. This funny looking machine here on the left is the Apollo guidance computer. It was developed at MIT for the Apollo shuttle launches. And it looks pretty old school, but it did amazing things. There were multiple Apollo missions here. On the bottom, that's Alan Shepard in 1971 on the Apollo 14 mission. This was a remarkable piece of technology, but to be honest, it's about as technologically advanced as a modern toaster oven. So all of the NASA Apollo missions were essentially launched with something with the technology of a toaster oven. Your iPhone today is millions of times more powerful than all of NASA's computing power in the 1960s or 1970s. And in fact, it wouldn't be wrong to say that your iPhone could have guided 120 million Apollo-era spacecraft to the moon all at the same time. So our ability to harness big data and integrate genetics, protein biomarkers, imaging, all sorts of other things is unparalleled. And that's the real evolution in medicine. And it's sort of this sphere of advanced analytics. And you already experienced this in other domains of your life, like Netflix. You would be shocked to know how Netflix predicts that because you watch some romance comedy that it says that you're going to love some zombie flick. And it turns out you do, because Netflix knows how when you stop it, when you scroll through the credits, it knows when you watch things, what time of day. It collects an enormous amount of information to help predict what your next movie purchase or show purchase might be. And that same sort of predictive analytics can be used from healthcare by integrating all of these different streams of big data. So using all of the information that's in your health system, in your electronic medical record, measuring all of the omics data, so all of the genetic material for all 3 billion of your base pairs, measuring your environmental exposures. It knows your zip code and all of the environmental allergens that are in your zip code, or what heavy metals are in the soil. It can measure things from like your smartwatch. So if you had an Apple Watch, it can measure your oxygen capacity based on your watch. And we're actually at the ability now where we can measure the microbiome in your gut, because we know that your gut bacteria is actually very important in determining whether people develop disease. So we can't just measure DNA. We can't just measure specific blood tests. We need to measure all of these things. And it's only now that we have the computational power that we can integrate all of them together to look for signals. So what does the future look like? Well, the good news is that we're currently actually pretty good at predicting your risk of having another blood clot. And it's pretty cheap and it's pretty accurate. And for the time being, at least for venous thromboembolism, it really doesn't incorporate measuring your genetic risk factors. It's not ready for prime time yet, although it will be in the near future. But I think we also have to take a step back and stop looking for the next best test. That's a flawed approach. What we really want to do is measure a personalized approach for each patient. All of you are unique. And so having a single test that we use in everybody makes no sense. We need to treat each person, each patient, as their own individual sort of test case, and then we need to measure everything for that individual patient. But to be honest, there's still a lot we don't know, and this is the famous Rumsfeld, the unknown unknowns. There's still, I spend a lot of time doing research in addition to clinical practice, and we have to be very honest that we're still learning a lot about why some people form clots and others don't, but we've come a long way in the last 10 years. But I'm very optimistic of the future, because I think the genetics revolution, as well as the emergence of a vast digital ecosystem in biomedicine, really puts us at an exciting precipice of discovery. So thank you for your attention. I'm happy to take any questions.
Thank you so much, Dr. Ruff. That was wonderful, really informative and helpful, and hopefully hope to, kind of calm a few fears that people had. We did get a bunch of questions in, so we'll dig right in.
Sure.
The first was, did I hear you right, that if I'm on lifetime warfarin, I cannot be tested again for factor V, Leiden, etc., because I will never go off warfarin?
Yeah, so I should a caveat is that some of the blood tests are affected whether you're on. So there's two parts to that question. One, if you're on a blood thinner, does it affect the testing? And that is true for most of them, except for the DNA-based tests, which are factor V Leiden and prothrombin gene mutation. So those, because they're DNA, you're actually measuring, they're sequencing the DNA. Those you could get an accurate result if you're on anticoagulation. So that's one part of it, that some of the tests can be done while you're on a blood thinner, most can't. The other question that I'm often get is someone will come to me on a blood thinner, they're referred, and they said, could you measure factor V Leiden or some other blood test? And if it's negative, take me off a blood thinner. And I would tell people who have that question, that's a bad idea, is that no one should use a genetic test at this time. We don't have enough information that says if someone had a negative screen for those 5 tests I mentioned, that their risk would be low enough that they shouldn't be on a blood thinner. So right now, the guidelines are based entirely on whether the clot is provoked or not. So if someone had a very good clinical factor for having a clot, they had major surgery, they took a 30-hour trip to Asia, they were on medications that increased their risk of having a clot, then those are called provoking factors. And if they had those provoking factors, it's reasonable, if they no longer have those factors present, to stop the blood thinner after they've completed treatment. But for people where you can't clearly identify a single strong provocative factor, that their recurrent risk is high enough, regardless of whether any of these 5 tests are positive, that it's not useful to measure it in those people because you'll give them wrong information. So if someone has an unprovoked clot, so not a real strong reason to say, oh, you're on this medicine, we know it causes clots, like say oral contraceptive pills. We know that causes clots in women. So mainly we say that we know that causes clots, we're just not going to give you OCPs anymore. Well, I don't need to test someone's genetic testing because I know once I stop the trigger, the OCPs, their risk is a lot less. They had a provoked VTE. But say I had another woman where I'm like, I really have no idea why you had a clot. Well, regardless of whether they had any of those tests or positive, their risk of having another clot is high enough that I would not use the results of that genetic testing to change the fact that that patient likely needs lifelong anticoagulation. So I would say that the people who actually qualify for genetic testing for VTE is probably well less than 5% of all of the patients in the United States of America who have a venous thromboembolic event.
Oh, okay, interesting. This question is kind of along the same lines. It's a little bit different. I was recently tested for von Willebrand's. However, I'm on Xarelto for factor V Leiden. Does being on the Xarelto mean the von Willebrand's disease test was not valid?
No, the von Willebrand factor is a separate issue. That's not an inherited thrombophilia. That's an acquired. So we didn't mention in this talk, but there are other conditions. We're talking about genetic tests for people who are born with variants that predispose them to disease. There are things that you can acquire that are not genetic that can increase your risk of having a clot. And von Willebrand's factor, along with antiphospholipid antibody syndrome, are two other things that you can acquire, but they're not genetically mediated. So these aren't, von Willebrand is not a test that I measured because it's not an inherited thrombophilia and it's not affected by any blood therapy.
Okay.
I will say that when we do test for von Willebrand's factor, we don't routinely give people blood thinners specifically for it unless they've had a clot. So if someone's had a clot and they happen to get tested for von Willebrand and it's positive, obviously they're on a blood thinner for the clot already. But if we happen to, for whatever reason, measure it in someone who never had a clot, I'm not sure why we would do that. It wouldn't be an indication to start a blood thinner in someone who's never had a clot.
Interesting. Okay, what are your thoughts on at-home genetic testing things such as 23andMe? Are the results accurate?
Yeah, it's a great question. The sort of direct consumer genetic testing, they're accurate in the sense of they are basically measuring some of the things that I told you. They'll give you a readout saying that you've inherited different variants that could predispose you or protect you from risk of all sorts of things: Alzheimer's, heart disease, cancer, breast cancer, et cetera, colon cancer. And so they're certainly accurate. The question is that, as kind of what I mentioned in the beginning, is that the information that they're telling you that says that you inherited these genes that put you at increased risk, the increased risk for most of the diseases they're testing you for is minimally increased. And that's the problem. It's not that they're inaccurate, but they might tell you that you've inherited these variants that increase your risk of having a heart attack. And so when people get that, they're like, oh my God, I'm going to develop a heart attack. It increases my risk by 80%. No, they increase your risk by maybe like 2%. And that's not really that much. And so that's the problem that I told you are the data that they're giving you is incomplete. You know, if someone told you, I'm going to give you a lottery ticket and your chance of winning is 1 in a million, you'd be like, well, I'm not that impressed. If I gave you a lottery ticket and you said that you had a 50-50 chance, of winning the lottery, that's a very different risk, right? Even though both of them technically increase your odds of becoming a billionaire. And so I'd say 23andMe is accurate, but it's not really that useful. You want to know where it's useful if you want to know your heritage. If you're, you want to know, what your lineage is, would your family migrated from in Europe or wherever. But I have to be honest, if you want to know, are you going to develop a disease or not, it's not really that helpful. Not at this time.
Is it helpful? I mean, if you say you were to do it and got, your results back that were concerning, I mean, is that something that you should discuss with your
Always,
healthcare
yeah.
provider? Yeah.
Always. I think it's very helpful to say if you do get the results back that tells you that you could be at risk for having disease, I would definitely counsel everybody to bring that information to your doctor and discuss it with them and to understand what that really means as far as your risk, whether there's any additional testing that you might do, whether there's any change in your treatment or management or prevention. So I think that that's a great idea. That I would say if one thing, first of all, I've seen a lot of people freak out when they get it because they feel like they're a ticking time bomb. And I would say that's definitely not true. Any of the things that you're getting back on this test, if they increase your risk, they do so minimally. But it's definitely still worthwhile to talk to your doctor and say, what exactly does this mean? Is there anything different that we should be doing as far as screening for something or testing for something else? So I think that's a great idea.
Mhm. Okay. Let's see. So, does the sex of a child influence how likely they are to receive the genetic predisposition to develop blood clots? And if not, can they nonetheless pass along this predisposition to their own children?
Yeah, it's a great question. There are some diseases, as I mentioned to you, there are 23 chromosomes. One's a sex chromosome. There are some diseases that are passed along on the sex chromosome, usually the X chromosome. And so there are certain diseases where they're more likely to occur in men or women, usually men. But the blood clot genes that we've been talking about, none of them are on the X or Y chromosome. And so there is no sort of genetic preference that any of those things are preferentially inherited by a boy or a girl. And so theoretically, if a parent does have one of those variants, they're equally likely to pass it on to a female or male offspring.
Hmm.
We
Interesting.
do know, for whatever reason, men are at higher risk for having blood clots in general than women.
Uh-huh.
Um, we don't totally understand why that is. It's thought probably that the hormones of the ovulatory cycles are protective in women. That's why an interesting thing, like for heart disease, men generally develop heart attacks 10 years before women.
Mm-hmm.
And women's risk doesn't approach men until they've reached menopause. And so
Huh.
we think that somehow that levels of estrogen and et cetera are somewhat protective for women. But again, that's not a genetic factor. It's just an environmental factor that these hormones in women are somewhat protective. And that's why we generally see men usually, for whatever reason, are at higher risk of having clots all over the place, lungs,
Hmm.
legs, heart, et cetera.
Interesting. So the acne and bad moods are worth it, I guess, to
Yeah,
some extent.
exactly. So you get a 10-year, women get a 10-year head start.
Awesome. All right, this one's a little bit more complex. Basically, this patient has had an idiopathic PE with a strong family history, but they've tested negative for every genetic possibility that their doctor tested them for. So she turned to online research, which led her to the results of the Edith trial. Are you familiar with that
A
trial?
little bit, yeah.
Okay, so her question is basically with factor 11 and this gene
Mhm.
RS2289252, why isn't it part of a genetic testing panel? Because the results of the EDIS trial show that this um, factor 11 polymorphism was significantly associated with VTE.
Yeah, and it turns out that that variant is very much like those other variants that I told you about. That it's weakly associated with VTE, and it has the same problem that all of those other tests have. In that, if you measure it in people who inherit blood clots, that it doesn't appreciably change your absolute risk of having a clot.
Mhm.
And we know in people who are completely deficient in factor 11, and so there are these rare cases where literally the gene is not present on either copy because you get two copies of either gene, that those patients don't seem to be at very high risk, even having 0% levels of factor 11 in the blood. They're not at high risk of having spontaneous blood clots, except in the setting or bleeding, except in the setting of having procedures. And so I think what this patient falls in that asked the question is there, if they have a strong family history, um, that it's a multiple first degree relatives at a young age, there likely is an inherited component. It is almost certainly not a single gene, whether it's one of those five that I mentioned on that, or factor 11, or there's a number of others. It's likely a combination of dozens or hundreds of genes. And we know this actually from heart disease work. A lot of work I do is in heart attack, and we have these families that we have multiple people where their family members died of a heart attack in their 30s and 40s, which is highly abnormal. And they don't have any of those very rare mutations like this patient asked for. What's that one gene that if I have it, my family is at risk for this clot? They don't have that one. But when we measure every single piece of DNA in their body and we sum up the hundreds of genes that we know that have it, they happen to get unlucky that 60 or 70 of those 100 happen to increase their risk. And their cumulative risk of having a clot is very high, but it's not due to any one single gene. And it's only by knowing that 100 of genes and what each one, because remember, each of those genes can be harmful or protective. You have to know each one and add them all up. I suspect in the person who asked this case that you could measure factor 11 is probably not going to help at all. And that it's only by measuring, and that's what people want. They want to measure one thing. But venous thromboembolism isn't generally a disease caused by one gene. That really, if you want to know someone's risk, you have to measure multiple genes. And so it's not a single blood test. It's basically you have to sequence all of those known genes. And we're still now, where I'm involved in one of them, a large genetic consortium in Europe, where we're trying to basically figure out what are the dozen, 2 dozen, 3 dozen genes that we need to be measuring in patients with venous thromboembolism. So we haven't yet defined that list, but everyone could name this one. What about this one gene? What about factor 11? What about X? All of them, if you just measure that one thing, is not going to tell you very much.
That's so interesting. And I think that probably has a lot to do with, when you hear things on the news, you hear about like scientists have discovered a gene that does this. So you just think that there's, all these genes in your body which individually have such a big impact on things.
You do, exactly, because we're conditioned upon. That's why I show that slide for these rare diseases, right? Everyone thinks that genetic diseases are all like cystic fibrosis, right? Where there's this one gene, and if you have this one gene, you're going to develop cystic fibrosis. And for cancer, you have this one bad gene, and you're going to develop breast cancer or pancreatic cancer, or one bad gene, and you're going to develop Alzheimer's. There are very rare diseases that's the case. And when we have those diseases, we can test family members and say, you got unlucky, you got that gene, or you don't have it, and you're not going to develop disease. But for most diseases, whether it's Alzheimer's, even breast cancer, although we have some information, but coronary disease, venous thromboembolism, atrial fibrillation, hypertension, diabetes, obesity, all of these things, they're not defined by one gene.
Yeah.
If they were, they would have been selected out by natural evolution. And so I think you have to take a step back and saying we have to stop trying to say, reading, the 2 lines of an article and saying, oh, I just need this one new test. All of these things, you need to find all of them. Like that's not to say that gene's not important or that factor 11 in this case is not important. It's just that in isolation, it's not helpful at all. So I need 100 factor 11 and I need to know all of them at one time for that one patient. So it's not as simple as people want it to be, but why should it be? Why should the human body be so simple? In this day and era of where we have this vast computational power, why can't we take full advantage? It's not a bad thing. We have all this information. We have all of this ability to measure. I can measure all your DNA. So why does it need to be just a single blood test?
Yeah.
And so I think it's just a lot more complicated than people think. But that's not a bad thing. It doesn't mean we won't get there. It just means we're not there right now.
Yeah. Very interesting. All right, one last question, and I think this is kind of an interesting question. Does a certain blood type, so A, B, AB, have any higher risk for blood clots?
Yeah, there's been a lot of research on this, and that there are some data that's conflicting that ABO blood typing can be associated with higher or lower risk, although it's been somewhat debunked. And so I'd say for the most part, in isolation, just whether you're AB or AB or O doesn't appreciably change someone's risk of having a blood clot. So it's not really useful to say, oh, you're O as opposed to your A. Your people's risk across blood types, although there was literature in the beginning about a decade ago to suggest it, it was likely due to other factors that we subsequently measured.
All right. Great. Well, we are about out of time. Thank you again, Dr. Ruff, for doing this. This was
My
incredibly
pleasure.
informative and interesting. If any of you guys have any other questions, you can email the info@natfonline.org email, and we will, forward those along to Dr.
Yeah,
Ruff.
please be happy to answer any questions. Thank you so much for your
Wonderful.
attention.
Yeah, we will also post a recording of this on the NATF website. So our next online support group is May 20th at 7 p.m. again, and we're going to be joined by Dr. John Grork, who is going to be teaching us about cancer and blood clots, so another important topic. So thanks again, Dr. Ruff, for joining us.
Thank you. Have a great day.
Yeah, thanks, and thanks for everyone joining us. Have a good night.
Bye-bye.